Welcome to this lecture on the comparative anatomy of vertebrates. Today, we are going to explore a truly fascinating topic, the evolution of urinogenital ducts. We will discover how nature solved a complex biological plumbing problem, ensuring that both waste removal and reproduction can happen efficiently and safely. Let us dive in. To understand this topic, imagine a growing city's underground plumbing system. Initially, when the city is small, there might be just one large, shared pipe carrying both drinking water and wastewater. As you can guess, this is chaotic and highly inefficient. Over time, as the city upgrades its infrastructure, engineers build separate, specialized pipes for different purposes to ensure safety and efficiency. In the vertebrate body, the internal plumbing system for waste removal and reproduction went through a very similar evolutionary upgrade. We call these biological tubes the urinogenital ducts. Now that we have our city plumbing analogy, let us look at the actual biology. The urinogenital ducts are specialized tubular structures responsible for transporting excretory products from the kidneys and reproductive cells from the gonads to the outside of the body. In comparative anatomy, we trace the evolution of these ducts back to a single shared ancestral tube known as the archinephric duct. This highlights a fundamental biological principle. As organisms become more complex, their internal structures compartmentalize to increase physiological efficiency. A shared tube works for simple organisms, but not for advanced ones. Building on that ancestral foundation, we see a dramatic shift in modern amniotes, such as mammals. These animals possess highly specialized, separate pathways. To truly understand this progression, we must look at how embryonic structures, specifically the Wolffian and Mullerian ducts, develop and differentiate across different vertebrate classes. During embryonic development, nature essentially remodels the plumbing. Ultimately, this evolutionary remodeling ensures that reproduction and excretion can occur simultaneously without interfering with one another. This separation is a massive evolutionary leap forward. This brings us to an important question. Why does the evolution of these ducts matter to us today? First, it explains the quirky anatomical setups we see in modern animals, including humans, such as why the male reproductive tract is so intimately tied to the urinary system. In medical science, understanding this shared embryonic origin is absolutely crucial for diagnosing and treating congenital defects. Furthermore, if we did not have this evolutionary separation, highly sensitive reproductive cells like sperm and eggs would be constantly exposed to toxic nitrogenous waste, drastically reducing fertility and species survival. Having established the biological importance, we must now look at the physics governing these changes. To truly understand why evolution favored certain duct sizes, we use Poiseuille's law. This formula calculates the resistance of fluid faces when traveling through a biological tube. In this equation, R represents the resistance to fluid flow. The Greek letter eta represents the dynamic viscosity, meaning how thick or sticky the fluid is. L stands for the length of the duct, and R represents the internal radius. Notice that resistance is inversely proportional to the fourth power of the radius. This means even a tiny decrease in the duct's radius will cause a massive, exponential increase in resistance, forcing the organism to adapt. Now that we have our formula, let us apply it to a concrete example. Consider an ancestral fish with a simple archinephric duct transporting dilute, watery urine. We are given a length of 0.1 meters, a radius of 0.002 meters, and a viscosity of 0.001 Pascal seconds. First, we calculate the radius to the fourth power, which gives us a very small number, highlighting its exponential impact. Next, we substitute our known variables into the equation. The numerator represents the resisting forces, and the denominator represents the space available. Solving this gives a final resistance of approximately 1.59 times 10 to the power of 7. Physically, this relatively low resistance means the fish's muscles do not have to work hard to expel urine. This baseline efficiency we just calculated leads us to a fascinating evolutionary problem. In a modern mammal, the ancestral duct evolved into the vas deferens to carry seminal fluid, which is 5 times thicker than urine, with a new viscosity of 0.005. To maintain the exact same energy expenditure, the resistance must remain constant. Therefore, the duct must logically become wider. To prove exactly how much wider, we start by rearranging Poiseuille's law to solve for the unknown radius. By multiplying both sides by the radius term and dividing by the resistance, we isolate the radius to the fourth power. This mathematical rearrangement perfectly mirrors the evolutionary pressure placed on the organism to adapt. Continuing with our derivation, we now plug in the new, higher viscosity of the seminal fluid and our target resistance into the rearranged equation. Notice that the numerator is now 5 times larger than in our previous example because the fluid is much thicker. This means the resulting value will also need to be larger to balance the equation. We are essentially calculating the new spatial requirement for the thicker biological fluid. When we divide the numerator by the denominator, we find that the radius raised to the fourth power equals 8.0 times 10 to the negative 11th power. We are one step away from finding the actual physical dimension. Now for the final step of our proof. The number we just calculated does not give us a physical dimension we can easily visualize. To find the actual physical radius of the new duct, we must take the fourth root of this number. Calculating this yields a new radius of approximately 0.003 meters, or 3 millimeters. Physically, this means the mammalian vas deferens must be about 50% wider than the ancestral duct to transport the thicker reproductive fluid without requiring extra energy. We have just proven mathematically how anatomical structures evolve in direct response to physical constraints. Having proven the mathematics behind duct expansion, let us look at the anatomical changes visually. On screen is a schematic showing the evolution of these ducts. At the top, we see the indifferent stage, representing the ancestral setup. Notice the shared archinephric duct in blue and the Müllerian duct in red, both leading to the cloaca. Now, look at the bottom left for male development. The archinephric duct is retained and modified for sperm transport from the testes, while the Müllerian duct degenerates. On the bottom right, in female development, the exact opposite happens. The Müllerian duct develops into the oviduct for eggs, and the archinephric duct degenerates. This beautifully illustrates the evolutionary bifurcation we discussed. This visual diagram perfectly reinforces our main theme. Nature acts as a master engineer. The evolution of urinogenital ducts is a beautiful example of nature repurposing existing structures to meet new biological demands. We started with a simple, shared duct that handled both waste and gametes. Through evolutionary time, this system bifurcated. Males largely retained and modified the ancestral duct for sperm transport, while females developed an entirely new tube for eggs. As we proved mathematically, these anatomical changes were not random. They were strictly guided by the physical laws of fluid dynamics to ensure maximum efficiency. To wrap up our lecture, let us review the key takeaways. First, we learned that urinogenital ducts evolved from a single shared ancestral tube into highly specialized separate pathways in modern vertebrates. Second, this separation is biologically crucial because it protects sensitive reproductive cells from toxic excretory waste. Third, we used Poiseuille's law to prove that anatomical evolution is constrained by physics. Thicker reproductive fluids mathematically require wider ducts to maintain energy efficiency. Finally, we saw how embryonic development beautifully mirrors this evolutionary bifurcation. You have now mastered the complex evolution of the vertebrate plumbing system. Great job!